Welcome back to the Mitosis. Today we're going to be making a cake for a special friend, Mr. Zhang. He's a biology teacher at the Bergen County Academy whose birthday is coming up. Alright, first we're going to get out our ingredients. Milk, eggs, organic. Alright, so you know what these eggs remind me of? Mitosis. Hey, we're already talking about biology. Why not talk about mitosis? So, let's pretend this here egg is one cell that's going to go through mitosis. It's just finished its G1, S, and G2 phases, and it's ready to divide again. So let's take a look at what mitosis looks like up close. So, let's pretend this egg is going through prophase, which would look kind of like this. Got the nucleus and the spindle fibers. So, as you can see, the DNA shortens and tightens up into rod-shaped chromosomes. As we know, all the chromosomes were already copied during the S phase, as two copies of each chromosome, the chromatids, stay connected to one another by the centromere. Cooking in science is so much fun. So anyway, at this time, the nucleus and the nuclear membrane break down and disappear. Then two paired dark spots called centrosomes appear next to the now disappearing nucleus. Whoops, I didn't have enough room on this egg to show that. Okay, so the centrosomes move toward the opposite poles of the cell. Then the spindle fibers made of microtubules radiate from the centrosomes. How cool. So this is called the mitotic spindle. Hard word. Polar fibers extend from the dividing cell, from the centrosome to centrosome, but don't attach to the chromosomes. Funky. Alright, we're going to be taking a short commercial break, but we'll see you in a bit, and we get to move on to the phase. How exciting. Welcome back. So, as I was saying, after prophase comes metaphase. It looks a little bit like this. Very cool. So, during metaphase, the second step of mitosis, the chromosomes are easier to be seen using a microscope. The kinetochore fibers move, so the chromosomes to the center of the dividing cell during metaphase. So as you can see, once in the center of the cell, the chromosome is held in place by the kinetochore fibers. I love cooking. After metaphase comes anaphase. Let's try that one. Alright. There we go. So, it's very similar, but as you can see, it's pulling apart. So during anaphase, the chromatids of each chromosome separate at the centromere and slowly move, centromere first, toward the opposite poles of the dividing cell. Cool. After they separate, they are considered to be individual chromosomes. Finally, we get to telophase. Telo? I was always more of a science kid. So, telophase, whatever. Anyway, during this, the chromosomes reach opposite ends of the cell, and the spindle fibers disassemble, so the chromosomes return to a less tightly coiled chromatin state. Okay, during telophase comes the actual division that forms the cleavage furrow, called, you guessed it, cytokinesis. Woo! Heavy stuff. So the last point I really want to make is something called control of cell division. Well, the control of cell division is very important because it presents very devastating things, such as cancer. Alright, thank you so much for watching my special science edition, Who Knew? And I'm going to get back to baking my cake. So, don't go yet, because we have a special episode next. The whole cast of Jersey Shore is coming in to talk about the massive amounts of dangerous radiation poisoning going on in that house. You do it all the time. Alright, see you soon.